Respiration is a general term for the exchange of gases between the human body and its immediate environment. Oxygen and carbon dioxide are two of the most important gases that are exchanged in the process of respiration. As we all know, oxygen is taken into the body in a process called inhalation, whereas carbon dioxide is sent out of the body in a process called exhalation. So this is respiration in general. We also have another important term which is called cellular respiration. So as the name itself implies, cellular refers to the cells in the human body. Cellular respiration is a set of chemical processes that take place inside the cells and utilize molecular oxygen to break down a sugar molecule called glucose and releasing the energy that is present in it. So this is cellular respiration. So cellular respiration can be defined as a chemical process that takes place in the body cells and utilizes oxygen to break down a sugar molecule called glucose and thereby releasing the energy that can be stored in a molecule called ATP. Now this is cellular respiration, whereas respiration in general mostly refers to the exchange of gases, or in simple terms it is also called breathing. Now cellular respiration is a key part of the broader life process of respiration, which is one of the 7 key life processes that are exhibited by all the living organisms. In this video, we will be studying cellular respiration in human beings and its significance. All living cells in the human body require energy. Now, before we move into studying cellular respiration, let us look at some of the important activities in the human body that are absolutely dependent on the energy that is released from glucose. So the first important process is contraction of muscles or muscle contraction. So as we all know, movement of our body requires the movement of muscles, or movement of certain portions of our body, as in our hands, also requires the movements of particular muscles, and for this the energy has to come from ATP. Next, so we all know that cells in our body undergo cell division. So cells divide on a regular basis, and this can be to either repair the damaged tissues, or it can also be to grow new cells. Again, cell division requires a lot of energy. Next is protein synthesis. So we know that proteins are one of the most important biomolecules that are there in the human body, and the synthesis of proteins requires a lot of energy. So ribosomes are the cell organelles at which proteins are synthesized, and protein synthesis as a whole requires a lot of energy. Next is active transport. Active transport at the cell membrane. So active transport refers to the movement of molecules against their concentration gradient. So we all know that diffusion helps to move the molecules from a region of high concentration to their low concentration. But if we have to do the opposite of this, meaning if we have to move molecules from a region of their low concentration to higher concentration, then this process requires energy. This is called active transport, and active transport is completely dependent on energy. So human beings are included under mammals, All the mammals and birds are essentially called homeotherms. They are homeotherms or warm-blooded animals. Warm-blooded animals. So we are able to maintain a constant internal body temperature, which is usually around 37 degrees centigrade. In order for the body to maintain this stable internal temperature, again it has to invest a lot of energy. So homeotherms or warm-blooded animals require energy so as to maintain a constant internal body temperature irrespective of their surroundings. Another example could be in the transmission of nerve impulses. Nerve impulses. So we know that neurons help to transmit the information from the brain or spinal cord to multiple tissues in the human body. So again, the transmission of nerve impulses across neurons also requires the investment of energy. And energy that is required for all of these processes essentially has to come from the breakdown of glucose. So these are some of the important metabolic processes that take place in the human body that is absolutely dependent on the energy that comes through cellular respiration, or in other words, through the breakdown of glucose. Energy for the body cells can be obtained by breaking down organic molecules such as glucose. So glucose is a simple sugar, or we can consider it as a monosaccharide, and glucose is an important carbohydrate. It is also an organic molecule. Nearly all of the energy for the living cells comes from the energy that is stored in the bonds of the sugar glucose. To understand, let us first look at the structure of glucose. The molecular formula of glucose is C6H12O6. So it has a total of 6 carbons. So this is the structure of glucose. So it has 6 carbons, carbon 1, 2, 3, 4, 5 and 6. So it has a total of 6 carbons, 6 oxygens, 1, 2, 3, 4, 5, 6, and a total of 12 hydrogens. Now this is the linear form of glucose. Glucose can exist in a linear form and also in a ring-shaped structure which resembles a hexagon. Okay, so let us study the open chain conformation of glucose. Now glucose is usually known as a polyhydroxyaldehyde. So if you look at the structure, glucose contains a lot of OH groups. OH here, here, here. So because it contains many hydroxyl groups, it is known as polyhydroxyaldehyde. So this is the functional group in glucose, which is an aldehyde. So CHO in chemistry refers to the functional group which is aldehyde. So glucose is commonly known as a polyhydroxyaldehyde because it contains multiple hydroxyl groups plus the functional group aldehyde. Now the potential energy in glucose is essentially stored in this carbon-carbon and carbon-hydrogen bonds. So the carbon-carbon and carbon-hydrogen bonds present in glucose contain a lot of energy. The potential energy is essentially stored in these bonds. In order to make use of this stored energy, the cells have to break down glucose. The cells have to break down glucose to release the energy. Now this is carried out in a series of chemical reactions that is termed cellular respiration. So the set of metabolic reactions through which the energy that is stored in these bonds can be released is referred to as cellular respiration. Like all the other metabolic reactions, cellular respiration also involves a lot of enzymes. So enzymes are basically protein molecules that help to accelerate the rate of chemical reactions. And cellular respiration occurs in the cells of all the living organisms, both autotrophs as well as heterotrophs. The complete process of cellular respiration involves 4 major steps. So the complete breakdown of glucose to release its stored energy takes place in 4 distinct steps, starting with glycolysis. So it's there in the name that glyco refers to sugar, which is glucose, and lysis is breaking down. So in glycolysis, the 6-carbon molecule glucose is broken down in a series of metabolic reactions to produce pyruvate. So pyruvate is a 3-carbon compound. 3C refers to the total number of carbons. Glucose has 6 carbons and pyruvate has 3 carbons. So this is the starting of cellular respiration. So conversion of glucose to pyruvate takes place in the cytoplasm or cytosol of the cells. Second, we have a transition reaction that converts pyruvate into acetyl coenzyme A. Coenzyme A is also written as CoA. So this is a transition reaction that converts pyruvate from glycolysis into acetyl coenzyme A. So this is an important cofactor. The next step is Krebs cycle, or it is also known as tricarboxylic acid cycle. Tricarboxylic acid. TCA is tricarboxylic acid, because the first stable product formed in this cycle is a 3-carbon compound. It is known as tricarboxylic acid. Krebs cycle takes place in the mitochondria. Glycolysis takes place in the cytoplasm, whereas Krebs cycle takes place in the mitochondria. The final is electron transport chain. Or oxidative phosphorylation. This is the final step of cellular respiration, and this is where much of the ATP is produced. And this process also takes place in the mitochondria. Okay. So glycolysis and Krebs cycle also produce ATP, but the amount produced here is minimal. The major amount of ATP is produced in the electron transport chain, which is the final step of cellular respiration. So the final output of cellular respiration would look like this. Glucose in the presence of oxygen So oxygen is absolutely essential for cellular respiration, and that's why this is also known as aerobic cellular respiration. Aerobic cellular respiration, because the process requires oxygen to produce carbon dioxide and water. So this is the final output for aerobic cellular respiration. So glucose is broken down in the presence of oxygen to give out carbon dioxide and water. So the carbon dioxide which is produced here is sent back to the lungs and it is exhaled out of the body. So carbon dioxide is a byproduct of cellular respiration and is eventually given out of the body through the lungs. Now in this process, glucose molecule is the one that gets oxidized, or it loses its electrons. So glucose is oxidized while oxygen gets reduced. So oxygen molecule gains electrons in the process and gets reduced into water. So oxygen is important in the whole process because oxygen is the final electron. Oxygen is the final electron acceptor. So the electrons that are lost from oxidation of glucose has to be taken up by oxygen, and in this process oxygen gets reduced into water. So oxygen is the final electron acceptor in aerobic cellular respiration. So this is the overall chemical reaction of respiration that involves 4 key steps. So in this video, let us discuss the first step of cellular respiration, which is glycolysis. So glycolysis is important because this is an ancient pathway that is observed in all the organisms. I'll be making a detailed video on all the steps in cellular respiration shortly. So let's study glycolysis. So glycolysis is a universal anaerobic pathway. It is anaerobic because the process of glycolysis doesn't require oxygen. It does not require oxygen, and hence glycolysis is anaerobic, and this takes place in the cytoplasm of all the cells. So glycolysis is a pretty ancient metabolic pathway that is observed in both the prokaryotic and eukaryotic cells. So in glycolysis, glucose molecule undergoes partial breakdown to form pyruvate. So pyruvate is a 3-carbon compound. So one molecule of glucose is broken down into 2 molecules of pyruvate. 2 molecules of pyruvate, which is a 3-carbon compound. This is the molecular formula of pyruvate, CH3CO COO minus. So pyruvate is a derivative of pyruvic acid. So if we add a hydrogen here, then it becomes pyruvic acid. It becomes pyruvic acid. It's important to note that glycolysis is a multi-step reaction and involves a lot of intermediate molecules. A lot of intermediates are produced in this process. So glycolysis to pyruvate takes place in a total of 10 steps that involves multiple enzymes. The final product of glycolysis is pyruvate. Now there are 2 important phases in glycolysis. 2 important phases. First, we have the energy investment phase and we have the energy payoff phase. So the entire breakdown process of glucose into pyruvate can be divided into an energy investment phase and an energy payoff phase. So as the name suggests, we are investing energy in the form of 2 molecules of ATP in this first phase. So here 2 molecules of ATP are basically consumed, and this helps to trap the glucose molecules within the cell. The glucose molecules which are taken into the cells need to be trapped so that glucose doesn't leave the cells again. And this process of trapping glucose requires 2 molecules of ATP. That's why this is known as the investment phase of glycolysis. So glucose gets converted into glucose 6-phosphate. So the glucose molecule essentially gets phosphorylated, and phosphorylated glucose molecule cannot leave the cells. So it is at the 6th carbon of glucose that phosphate group is added. So we call it as glucose 6-phosphate. So glucose is phosphorylated. In the next phase, which is called the payoff phase, here 4 molecules of ATP are generated. The energy payoff phase helps to produce 4 molecules of ATP. In addition, the electrons that are released from the breakdown of glucose is transferred to NAD+. So NAD+ is an important coenzyme in cells, and it helps to power most of the cellular processes. NAD+ is nicotinamide adenine dinucleotide. So like we said, the glucose molecule gets oxidized, and the electrons that are lost from glucose gets transferred to NAD+, which is an important coenzyme. So NAD+ can actually exist in 2 different forms. So we have NAD+. NAD+ can accept electrons and hydrogens and can get reduced into NADH. This is the reaction that is taking place in glycolysis. So NAD+ accepts the high energy electrons from glucose, and NAD+ gets reduced. So NADH is considered the reduced form, whereas NAD+ is the oxidized form. It is also possible for NADH to lose the electrons and then get converted to NAD+. So this is essentially a reversible reaction. NAD+ by accepting electrons can get converted or reduced to NADH. NADH can lose the hydrogen and electrons to get oxidized back to NAD+. So this forward reaction is the one that is taking place in glycolysis. This electron transfer process actually lies at the heart of cellular energy production. Okay, so NAD+ is a electron carrier. The net reaction of glycolysis would look something like this. So glucose C6H12O6 plus 2 molecules of ADP plus inorganic phosphate plus gets converted into 2 molecules of pyruvate. C3H4O3 is pyruvate plus water. So ADP is getting 2 molecules of ADP are getting 2 inorganic phosphates. So we get 2 molecules of ATP, and NAD+ is getting reduced into NADH plus 2 molecules of NADH plus 2H+. So glucose gets broken down into 2 molecules of pyruvate. Now if you remember the energy investment phase, it utilized 2 molecules of ATP. It utilized 2 molecules of ATP, whereas the payoff phase actually produced 4 molecules of ATP. Because 2 molecules of ATP are itself consumed in the process of glycolysis, the net production of ATP is only 2 molecules. Additionally, 2 molecules of NAD plus are reduced into NADH, which is its reduced form. So 1 molecule of glucose through glycolysis will produce 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. Now pyruvate that is produced here moves to the next stage of aerobic cellular respiration. As we remember, there is a transition step that converts pyruvate into acetyl coenzyme A, and acetyl coenzyme A will next go into the Krebs cycle. This is the fate of pyruvate. The 2 molecules of ATP can be released into the cell for using the energy available in ATP. Now NADH has 2 possible routes. NADH produced here can travel to the mitochondria, carrying the high energy electrons to electron transport chain. So this is one possibility for NADH, or NADH can also be used to regenerate NAD+. So NADH can also be used to regenerate NAD+ through a process called fermentation. Fermentation that either involves production of lactic acid or ethanol can help to regenerate NAD+, and NAD+ can again be fed into glycolysis. NAD+ can again be utilized by glycolysis to regenerate NADH. So these are the fates for the molecules produced in glycolysis. Now to conclude, let us look at some of the important functions of glycolysis. So glycolysis takes place in the cells of all the organisms from prokaryotes to eukaryotes, and glycolysis helps to produce some energy. So 2 molecules of ATP are generated in the process of glycolysis, so some amount of energy gets trapped here. Second, glycolysis is very important for red blood cells as it is the only source of energy production. Red blood cells do not contain mitochondria. So as they lack mitochondria, they cannot carry out Krebs cycle or electron transport chain. So RBC is completely dependent on glycolysis to produce some energy. Glycolysis is very important in hypoxic cells. In active muscle cells, where oxygen is limiting, glycolysis is the only route for production of ATP. So glycolysis is the only route through which some molecules of ATP could be produced. So this is a basic overview of aerobic cellular respiration and glycolysis in particular. So I'll be making a detailed video on the complete processes in cellular respiration shortly. Moving on, apart from aerobic cellular respiration, we also have something which is known as anaerobic respiration. Anaerobic respiration. And as the name implies, this is the form of respiration that doesn't require the presence of oxygen. Aerobic refers to oxygen, whereas anaerobic refers to absence of oxygen. Production of ATP can also take place through other modes in some organisms. So one of it is anaerobic respiration. The common thing between aerobic and anaerobic respiration is that both of them involve an electron transport chain. So electron transport chain is the final step in aerobic cellular respiration. Anaerobic respiration is also similar in that it also employs an electron transport chain. But the final acceptor of electrons here is not oxygen. Because oxygen is not available for these organisms. With anaerobic mode of respiration, the electron transport chain is still there, but the electron acceptor is something other than molecular oxygen. So some compounds that can accept the electrons in anaerobic respiration include sulphur, sulphate, SO4 2-, and also nitrate NO3 2-. So these are some of the molecules that can act as the terminal electron acceptors to generate ATP. Anaerobic respiration is usually observed in prokaryotes like archaea and certain types of bacteria. So these are two species in which anaerobic cellular respiration takes place. And most of these organisms are usually living in low oxygen environment. So they don't have access to molecular oxygen. In the absence of oxygen, they need to switch to other molecules that can act as electron acceptors. Some of the bacteria can use sulphate as the terminal electron acceptor, and they produce hydrogen sulfide as the end product instead of water. In the case of aerobic cellular respiration, oxygen is the terminal electron acceptor and oxygen gets converted to water. However, in some bacteria, sulfate accepts electrons and gets converted into hydrogen sulfide. Some species of archaea use carbon dioxide to produce a byproduct which is known as methane. So methane is CH4. So these organisms use carbon dioxide to produce methane. So these organisms are usually found in the digestive system of ruminants like cow, where they help the organisms to digest cellulose and in the process releasing methane as a byproduct. Now the last category is fermentation. So fermentation is also a type of process where some amount of ATP can be generated. Fermentation is again divided in two categories. First, we have lactic acid fermentation and alcohol fermentation. Now, unlike aerobic and anaerobic cellular respiration, fermentation actually doesn't involve respiration of any kind. So we don't observe an electron transport chain in fermentation, while this is observed in both aerobic and anaerobic cellular respiration. In both the forms of fermentation, glycolysis is the first step. So breakdown of glucose to produce pyruvate is the initial step in both the forms of fermentation. Now glycolysis is an ancient pathway and is observed in all the organisms. So depending on the fate of pyruvate, we have two different types of fermentation, which could be lactic acid or alcohol fermentation. Let's start with alcohol fermentation. Alcohol fermentation is commonly observed in the brewer's yeast. Brewer's yeast is a common name for the organism Saccharomyces cerevisiae. So Saccharomyces cerevisiae is an important unicellular yeast that belongs to the kingdom fungi. It is a unicellular organism and it can undergo alcohol fermentation. The first step here is again the conversion of glucose to 2 molecules of pyruvate. So we have 2 molecules of pyruvate, which is a 3-carbon compound, CH3. This is the structure of pyruvate. Now pyruvate undergoes a decarboxylation reaction. Decarboxylation because it is losing a molecule of carbon dioxide. Decarboxylation to produce 2 molecules of acetaldehyde. CH3CHO. So acetaldehyde contains a functional group that is aldehyde and its molecular formula is CH3CHO. Now acetaldehyde is the electron acceptor as it accepts electrons from NADH and gets converted into ethanol. Which is C2H5OH. And in this process, NADH gets converted back into its oxidized form, which is NAD+. So pyruvate, which is a 3-carbon molecule and produced through glycolysis, undergoes a decarboxylation reaction producing acetaldehyde, which is an aldehyde. Acetaldehyde can accept electrons from NADH, thereby producing ethanol or ethyl alcohol. And in this process, it is able to regenerate NAD+. So glycolysis requires the presence of NAD+ to continue. This reaction is especially important in the brewing industry as this helps to produce ethanol. The same yeast is also extensively used in the baking industry because the carbon dioxide which is released in this process is what actually causes the bread to rise during baking. Alcohol fermentation has 2 big advantages to the industry. One is through the generation of ethanol, which is routinely employed in the brewing industry as in the preparation of beers and wine. Second is in the baking industry where carbon dioxide helps the bread to rise. The other form of fermentation is called lactic acid fermentation. The muscle cells in the human body and bacteria, namely lactic acid bacteria, which is also known as LAB, stands for lactic acid bacteria, predominantly use this mode of fermentation to produce lactate. Similar to alcohol fermentation, this also takes place in 2 steps, starting with glycolysis. So in glycolysis, glucose gets broken down into pyruvate. So let's start with pyruvate. Here, pyruvate itself accepts the electrons from NADH and gets converted into lactate. C3H6O3. Again, NADH loses its electrons to pyruvate, thereby regenerating NAD+ back. Okay, so we don't have a decarboxylation step here. The pyruvate produced through glycolysis itself acts as an electron acceptor, accepting the high energy electrons from NADH and gets reduced into lactate. Now this step is again crucial as it allows NAD+ to be regenerated, thereby allowing glycolysis to continue and produce some molecules of ATP. So both the forms of fermentation are actually helping to regenerate this important electron carrier, which is NAD+. Whenever we are doing a vigorous physical activity like running a marathon or even a short sprint, the muscle cells will also carry out this mode of fermentation as oxygen becomes limiting in these conditions. The body is not able to get as much oxygen as it requires, and in these conditions the body or the muscle cells will also temporarily switch to producing lactic acid. So some amount of ATP will be released here, and that will be utilized by the muscles to carry out its further work. Now what do you think will happen to the lactate that actually builds up in the muscles during this activity? Pause for a moment and think what could be the fate of lactate produced in the muscle cells. The lactate actually gets converted back into pyruvate. So pyruvate is a 3-carbon compound, and this pyruvate can again be fed into aerobic cellular respiration. So production of lactate is just a short-term process, and whatever lactate gets produced travels to the liver where it gets converted back into pyruvate. Now we know pyruvate can then enter into the next phase of cellular respiration, which is Krebs cycle. So to conclude, we have aerobic respiration, we have anaerobic respiration, and we also have fermentation. In aerobic and anaerobic cellular respiration, it starts with glycolysis. Then we have the Krebs cycle or TCA cycle and the electron transport chain. So all these events are common to both aerobic and anaerobic respiration. The only difference being oxygen acts as the terminal electron acceptor here, whereas sulphur or sulphate can act as the terminal electron acceptor for anaerobic cellular respiration. Now fermentation is different because it only involves glycolysis from the above. Only glycolysis takes place in fermentation, plus the additional steps that take place is only to regenerate NAD+, so that it allows glycolysis to further operate. So this is a basic overview of the different types of cellular respiration that can take place in different organisms so as to produce ATP, which is the universal energy carrier. If you found this video helpful, please do share it with your friends.